Cancer is one of the most potent diseases, affecting over 1 in 3 people across the globe. Particularly, breast cancer accounts for about 30% of all female-related cancers. Although it is prevalent in women over the age of 50, younger victims of breast cancers are on the rise. What makes it particularly dangerous is the ability for the symptoms to go unnoticed in the earlier stages, leading to metastasis and ultimately death. Recent studies have tried to find link between tamoxifen and a transcriptional co-regulator NUPR1. Tamoxifen is a hormone therapy drug aiding in prevention of metastasis and regulating estrogen levels to reduce chances of return of breast cancer. NUPR1 is a transcriptional co-regulator that converts stress signals into a gene expression, providing cell with resistance against the stress factors, in this case tamoxifen. Mode of action of tamoxifen. Tamoxifen acts as a competitive inhibitor of estrogen, blocking receptors and preventing tumor cells from using estrogen to spread. Tamoxifen causes the levels of tumor growth factor, alpha and insulin growth factor 1 levels to fall, while raising the levels estrogen-binding globulin. This decreases the overall availability of free estradiol, preventing tumor growth. As a further mechanism, tamoxifen is inhibitory toward protein kinase C, leading to cell death of estrogen-positive cancer cells. The alkylaminoethane side chain is important for the anti-estrogenic activity. The complementary hydroxyl group present will have high binding affinity. Para-hydroxylation of carbon-1 can lead to a greater potency drug. Experiment. With extensive use of hormone therapy, there is a high chance of resistance developing over time. A set of experiments were conducted to establish a relationship between NUPR1 and the possibility of combating TAM resistance by various mediated transcriptional methods to regulate target genes. For this, a TAM-resistant, tamer cells were cloned from estrogen receptor alpha-positive breast cancer cell lines MCF7, T47D and BT474 by a 15-month exposure to TAM. The analysis through assays and RT-PCR were conducted on these cell lines, and here are a few of the relevant findings. Results. 1. NUPR1 depletion overcomes TAM resistance in vitro and in vivo. According to the experiment, the results indicated toward the depletion of NUPR1 was enough to induce a G0/G1 arrest of the cell cycle. The proportion of cells in S phase in the tamer cell line were low despite only 0.4 of TAM being added. As a consequence of the depletion of NUPR1, there was a loss in a senescence-specific marker P21 and an increase in cell cycle inhibitors P16 and P27, confirming the role of NUPR1 in maintenance of cell cycle. In the athymic nude mouse model, the TAM treatment reduced the volume of tumor formation in the mammary fat pad. The knockdown of NUPR1 co-regulator reduced the proliferating nuclear antigen-positive cells, negating the effects of TAM resistance and thereby effectively overcoming breast cancer. 2. NUPR1 physically interacts with ESR1. An experiment using mass spectrometry and affinity purification was conducted to identify the proteins associated with the flag-tagged NUPR1 cells. A complex was recognized formed between NUPR1 and ESR1, indicating it a functional regulation role. ESR1 functions by recruiting transcriptional coactivators or corepressors to mediate endocrine resistance at regulatory regions present in the target genes. A series of assays like immunofluorescence and co-immunoprecipitation were conducted to establish the association. As you can see in the image, the localization was seen predominantly in the nucleus as compared to the cytosol. The co-IP was conducted with TAM treatment and the results were consistent with the other assays, indicating it a complex. To further this, a deletion assay pointed toward the need for a nuclear localization signal in order for effective binding between NUPR1 and ESR1. 3. NUPR1 regulated gene signature is used as a predictive for endocrine resistance. A transcriptional profile was generated by RNA sequencing to determine the effect on the target genes upon NUPR1 depletion. The results of the KEGG pathway analysis indicated towards the depletion of NUPR1, which further affects the estrogen signaling pathway, cell cycle and lysosomal processes. One interesting observation is the downregulation of drug resistance gene. The depletion of NUPR1 in MCF7 tamer cells led to a decrease in interleukin-6, also known as the pro-migratory cytokine. This further indicates the role of NUPR1 in induction of IL-6 in TAM-resistant breast cancer cells, hence confirming that in endocrinal therapy resistance, NUPR1 mediates autophagic survival by mediating a transcriptional control. To evaluate and conclude these findings, we can look into a few things and summarize our findings. Although there have been several breakthroughs in anti-estrogen therapies for patients with ESR1-positive breast cancer, very advanced stages of it are still incurable. A top factor for this being the resistance developed for endocrine therapy and the chances of reoccurrence. Here, we established that the co-regulator NUPR1 utilizes ESR1 to induce autophagic survival in response to TAM treatment in TAM-resistant cells. However, there was no significant relation between NUPR1 and breast cancer subtypes. The depletion of the co-regulator encouraged autophagic degradation, leading to reduction in malignancy and premature senescence. This suggests NUPR1 is an important factor in developing TAM resistance, promoting a higher lysosomal state in breast cancer cells via ESR1-mediated transcription. The levels of NUPR1 are at a higher level in populations of endocrine-resistant breast cancer cells, and the depletion of NUPR1 in TAM-resistant cells affected their survival severely when treated with tamoxifen. In addition to the interaction between ESR1 cells and NUPR1, it can regulate epigenetic modulators. As an extension to this thought, trans-epigenetic remodeling has been tested in diabetic mice using the CRISPR/deficient Cas9 system. These endogenous transcription regulation methods are promising in developing targeted drug resistance to cancer therapies. Additionally, the definition of tumor resistance is selection of resistant clones and the acquired homeostatic resistance. However, distinguishing them is almost impossible due to tumor heterogeneity. The signaling pathways and autophagic processes are controlled by millions of signals and transcriptional regulation which are very difficult to detect and alter. Upon treatment with TAM, the levels of NUPR1 was increased as an adaptation response to increased autophagy to protect cells from death. NUPR1-mediated transcription is responsible for the control of premature senescence. Senescence is considered an integral part of tumor development and therapeutics. Targeting NUPR1-mediated autophagic regulation can prove beneficial for making cancer cells more sensitive to anti-estrogen therapy. Therefore, an appropriate conclusion would be that there needs to be more research into TAM and small molecule inhibitors of the autosomal pathway to prevent recurrence of breast cancer.